We good to go? All right. So for those of you who don't know me, my name is Sophie Kearney, and I'm a summer intern in Dushan's lab. And I'm actually from Waco, Texas. So I go to school at Baylor University, and my major is bioinformatics. So I do a lot of the biology and the computer science side. And so my project for this summer is to create an ImageJ plugin that will perform nuclei segmentation and DNA damage classification. So let's get into it. All right, so the input for this plugin is actually a multi-image TIFF file. And so there's 3 different images that are included. And they're each stained with a different type of stain that will reveal different information about the tissue. So the first stain that we have is the DOPI stain, and DOPI is actually a small molecule that is attached to DNA. And so the purpose of this stain is to be able to actually see the different nuclei that we're seeing in this tissue. And so in this image, we have about like 3,000 different nuclei, and you can see each of those little circles is showing that nuclei. And so the usefulness of this image is that we can do nuclei segmentation and identify the exact coordinates, you could say, of the image where the nuclei are for further processing when we're using the Ki67 stain and the H2AX stain, which won't always fluoresce for every single nuclei. So with this DOPI stain, we can identify where all the nuclei are and use that in our future analysis. And the next stain that we have is this Ki67 stain. And so what Ki67 is it's actually a protein that's used in cell cycle proliferation. And so when the cell cycle is active, like in any stage besides G0, which is a dormant stage, Ki67 will be present. And because it is fluorescent in this stain, we're able to see which nuclei are in active cell cycles. And so that's the purpose of this stain. And so as you can see, going from this image, which has all the nuclei, to this image, which only has some of the nuclei, we're able to identify which ones are specifically in an active cell cycle stage. So the next stain that we have is this H2AX stain. And again, it's not all of the nuclei are actually shown, only some of them are fluorescent. And that's because the H2AX stain, H2AX is actually a modified histone. And so the histone binds to double-stranded breaks in DNA. And so here we're seeing severe DNA damage in the nuclei that are lighting up. And so this is a way that we can identify which nuclei actually have damaged DNA. And so that's how you can see some of these are fluorescent. And so usually biologists, when they're doing all of these stains, they're going to have to hand count everything, and especially get some ground truth in order to train models. They would have to sit down and hand count all of the nuclei that are positive or negative for H2AX. And so for each of these stains, for Ki67 and H2AX, if it is positive, that means that they are fluorescent in the nuclei. And so that means for H2AX, if it's positive, that means there is DNA damage in that nuclei. If it's negative, there isn't DNA damage above a certain threshold. And the same for Ki67. If it is positive, it means that nuclei is more fluorescent. And so that means that that nuclei is in the active cell cycle stage, and it's not in that dormant G0 stage. So we have these 3 different images, which are contained in that TIFF file, which are inputted into this ImageJ plugin. So yeah. All right. So the next stage is we're actually getting into the analysis using different models that Jotherson has created, which is amazing. So I didn't have to write them, but I'm just implementing them into this model. And so the first stage is that nuclei segmentation stage. So as you can see, this picture is very busy, but there's boundary boxes that are drawn around each of the nuclei. And so we're able to identify them using a faster RCNN network. And so it's able to identify using a rotation trick. So it essentially takes the part where the nuclei is detected and it rotates it to find the boundaries. And then we've drawn those boundary boxes around the nuclei. So in this image, we detected 3,000 different nuclei. And so we're able to use these coordinates in our future analysis. So we don't have to just search every part of the image. So we get the nuclei segmentation. We have all of the boundary boxes where we've detected these nuclei. And the next step is this Ki67 classification analysis and H2AX classification analysis. So we're going back to this Ki67 image. And so what we've done again is we're highlighting here in the boundary boxes the nuclei that were determined to be positive Ki67. And that means again that they're in an active cell cycle stage. And so the boxes that are shown here, even though we see a lot of different nuclei here, the ones that are positive were determined to or the ones that are drawn were determined to be positive, which means they're in an active cycle. So we're able to determine biologically relevant information using these models. So we can see right here, kind of zooming in here, we can see there's a very clear like nuclei kind of in the, I don't know if I can point. Oh, right here, right? We can see this nuclei, but it didn't pass that threshold. And so there's not a boundary box around it. So that means that it would be a negative. But then right here, we can see these really clear regions where there's a lot of fluorescence. And so these were identified as being positive for K67. All right. Here we go for the H2AX image. Again, we have the same boundary boxes drawn around where it's positive. And so when we zoom in here, we can see like this is a very fluorescent cell, and we have lots of nuclei. We have these other nuclei which weren't as fluorescent, so they weren't labeled as having DNA damage. All right, so the next step is this TSNE analysis. And what TSNE does is it's kind of collapsing our multidimensional data into a lower dimensionality. So we're able to visualize kind of the relationships and identify clusters and similar features that are shown in our data. And so it's a good way to visualize what's actually happening and identify patterns, especially when we're trying to determine if, our nuclei that have this DNA damage, are they similar to each other, or are they more similar to the nuclei that don't have the damage? We're trying to identify that there is kind of a clustering pattern in the similarity between the different classifications that we have between positive or negative H2AX or Ki67. So now we're getting into the output of this ImageJ plugin. So we do all of this in the boxes. That's all of the analysis that we're running. And then the circles on the left-hand side are our input. And then we have our output. So our first output is the TSNE interactive visualization. And so theoretically, it will be interactive. So you can kind of hover over and you can identify where in the image you have those regions of DNA damage or active cell cycle nuclei. And so we'll kind of look into that in the demo. And then our other output is those annotated images with the boundary boxes. So this is our final kind of pipeline of the input and the output. And then we can go into the demo. So this is all written in Java, which ImageJ is written in Java. And so they actually have a plugin that you can use that kind of provides a lot of the different like uses you would need. So I'm going to run it right now. We get to pull up ImageJ, and immediately we're going to choose this TIFF file. This is just a randomly chosen TIFF file, but it was in the slides, so that's why I included it. So it pulls up the image right here, and then we're going to go to this next part, which is asking for the folder for the output. And so I'm just going to choose this folder. And I ran the results before because it takes about a minute to run, so I didn't want us to just be sitting here. So fair warning, it's already been run. And then for this file right here, we don't actually have the DOPI model like running right now, so it's not integrated into this pipeline, but that's definitely a future step. So we're just inputting the code this would be the output of that DOPI model, and so we're just inputting it right here. But in a future step, we would have it wouldn't need to add this results file. Okay, so running the analysis, and then it would normally take a little bit longer than that. And so here are our results. So we have, before size filtering cells, we have 3,478. I'm not going to read all those numbers because that seems boring. Okay, after size filtering, we have around 2,700. And then here we can see our positive and negative Ki67. And so it's going through these size filtered cells and identifying the positive and the negative nuclei. Okay. And so it's pulling up these images with the boundary boxes while we're running that TSNE. And so as you can see here, these aren't as bright because they haven't, you can always adjust the brightness, but it's pulling these up as ImageJ images while we're running this TSNE. And perfect. All right, so it pulls up the TSNE as an HTML file because it makes it a little bit easier to run the different hover tools that we're going to be making in the future, which are partially made right now. So on this right-hand side, this is the original image. That's the DOPI image. And then we have K67 and then the H2AX image. And then on this left-hand side, we have this TSNE projection. And so right now, because this is only on one image, it's just kind of a circle. But if you're doing more of an aggregated with several different images, it won't just look like a circle. But the good part is that we do actually have clustering with these one labeled, which actually means that it's a positive K67. So this is just for the K67 image. We can also run this on H2AX. We just haven't done that yet. And so it's pretty good that we have this kind of cluster of K67 positive, and then the rest are K67 negative. And as you can see, as I scroll over, theoretically you'd be able to see the nuclei there, but we're working on that. That is in process. And then as you would scroll over a specific point, it would also in the future highlight where on this image it is. So this is kind of still being developed, but this is kind of a rough draft of what we're making. So yeah, and you can kind of scroll around here. It's pretty cool. So that is the input and the output of the program that we have. And again, it's all written in Java, and then it's run Python scripts to run all of those models. Okay. So the next steps that we have are to meet with our users. So this is being created for people who don't necessarily have all of that coding experience. And because they are familiar with ImageJ, it would be a lot easier to integrate into the workflow of a biology lab. And so that's kind of the point of creating this plugin. And so we're hoping to meet with those users and identify areas where we can improve this model, or improve this plugin, make it easier for them to use, and maybe add on some features that they would want to incorporate. And then again, running it on a directory of images so we can get the aggregated TSNE, and also not just do one image at a time, but do several images at a time. And then again, integrate the DOPI analysis that is in progress, and then finish the TSNE interactive graph, and then package and deploy the plugins. Right now it's running locally, but I would like to, I don't think it's going to be very difficult to do that, but that's kind of the next steps, is just making sure that we can distribute it to people and it's usable. So does anyone have any questions? It's pretty quick, but you know, I'm doing one project right now.
Yeah, I think it looks great. Any questions?
So the thing is running on the image pixels based on some representation from the model that you get.
It's running on the features that are outputted from the model. So that's, I'm not totally sure what all of those are, but that's what it is running on. Yeah, he would know more about this. Oh, Jyotishan, do you want to talk about the, I don't know if you heard the questions. Okay.
Can you repeat the question? Like I didn't hear it.
Ah, here we go. All right, sorry, go ahead. Okay.
Yeah, I didn't quite hear the question.
I was curious, like TSNE on which representation it's running. So Sophie mentioned that it's running on the representation from the, I think, fast RC, and then
No, actually, there's transit like so after detecting the cells, so we take the Ki67 or like the H2X images, and we do something like a representation learning on top on that, like using the SimCLR V2. So in that, like after training encoder on it, like we use those representation to do the classification as well as to do the TSNE analysis.
right. Thank you.
So what the tool, can you share your screen?
Yeah.
Yes.
What did you want me to share?
The demo maybe.
Oh my goodness, yes, sorry. Yes.
So you guys will be cut. So I think the TSNE is like TSN results of the TSN analysis is like a web tool, right?
Yes.
How you guys are thinking about it, like so you run it through ImageJ and the results will be like on a web page.
So it actually creates an HTML file.
Okay.
So this is similar to the code that Josh had before for his visualization tool. So it's not like, it's just a local file.
I see, yeah.
Yeah.
So, no, do you think like you can integrate that into the image?
Yeah, you
Because it's-
can,
Oh, is that too difficult?
no, I was looking into that. I kind of had it like this, kind of, we're in development, but you can actually integrate an HTML file. I think if we wanted to keep this, like interactive part, I think it's easier to do in HTML because you can run JavaScript, in HTML, although I can look into it, but what we could do is we could create this HTML file and then display it in the image shape plugin. There's some packages that do support that.
I see, I see. Yeah, the other option could be actually, there might be already plugins that actually do TSNE and
Yeah,
might also not looking a little bit like if there is anything that does exactly the same thing in the image ecosystem.
okay, yeah, I can look into that, for sure.
And in terms of the timeline, like when do you think we can like reach out to our collaborators and then show them what we have done and get some feedback?
I'm hoping to have a lot of like the next steps. It kind of depends on at what stage we think it would be good for them to see. Because I mean, right now we have like kind of the basics of it, but I'm hoping in this week to do a lot of those future steps I was talking about, and I can work on this TSNE and the like making it run on the directory of images is not going to take very long. So like I could have that done in like the next couple of days, and so.
Yeah, so I'm thinking like, so we also have to look at your timeline, right?
Yeah.
So, and also like after the meeting, you'll need some time to kind of like work
Yeah.
on whatever the comments you made.
Mhm.
So maybe I will reach out to them
Yeah.
like today and then try to schedule a meeting for early next week.
That sounds good, yeah. Cause I'll be here until the 11th, so
Correct?
that sounds good.
Alright, sounds good. And okay. And I think like in terms of your presentation, I think it's also good like if you can go through the manuscript Jatushan and Priscilla's
Yeah,
written and kind of like get an idea about how the models are trained, what kind of models are used, stuff like that.
Mhm.
Yeah, you don't have to like do it, but like it's good to know. Yeah.
yeah, I did abstract a lot of those details because I wasn't totally solid on all of them, but I do agree that, like, I read the script, I was like, oh, that's how that works, now I get it, so I think it'd be good to incorporate those details, especially like the slim CLR thing that you mentioned, I knew that, but I had forgotten to mention it, so I think it'd be good to include that.
Sounds good. And what do you think in terms of like, do you want to try training some of this or like running some of these models, get some idea on how they work and stuff like that?
I think I would enjoy doing that, yeah. I wanna make sure I can get this done as well, but I think it'd be fun to do some more of that side of.
Yeah, I think like the tool by itself, it seems like it's sort of like coming together. So I wouldn't worry too much about it, like completing that part. But I think like it's also good to kind of like know the background of it and
Yeah.
get some experience on like training of the models.
Yeah,
Or like at least like running inference on it,
Mhm.
on one of the models. Yeah. Alright, sounds good. Jatushan, do you have anything to add in terms of what Sophie has done so far and how it can fit into the paper?
So this one so there's only one. I think Sophie has already mentioned in the future work, the parts which have to be completed, like the problem we have with the DARPy detector. So I think kind of like Crusader is looking into that. The another part what we should include is so this like for labs like Bevin, she can use it to like do the inference on the images here. But like in terms of some other biological labs like which require to train on a different kind of images, so we might have. What do you think about like including a feature to enable the user to train the model itself? without like having the complexity of the code, and then
Yeah.
Yeah.
Yeah, I think it would be a good idea, but then like you can't run it on a thin client, right? Like
Yeah.
you can't run it on your laptop, so you need to sort of figure out all the other backend stuff, right?
Yeah.
Where the data is gonna be, like all that. So have you given a thought about like how that will come to it?
Yeah, Darshan, like I think that would be a bit of complexity because like without a laptop or like a device with a GPU, it would be hard to train on the edge itself. Yeah.
Yeah, unless you have like a pre-trained model that can kind of like quickly like
Fine tune. Yeah.
fine tune on a smaller data set. The other option is like we need to sort of link in some some way to like communicate with the cluster or a given cluster.
Yes.
Yeah, but I think like for that part it would require some technical like knowledge, right? So I don't see actually that at least David's group doing that by themselves.
Yeah, so like that would be like bit of a tough. So maybe like we can like with the paper like we can provide the code in the GitHub for like to reproduce it, but like even if it's a biology lab want to do so, they might have to have someone in the technical side to run it. Yeah, if they want, if it's required to like train the models. Yeah, but in terms of inference, I don't think it would be an issue. Yeah.
Yeah, I think this would mostly be on the inference. That's what I was also thinking.
Yeah.
But it might be good if you can demonstrate it on a different data set as well.
My secondary mentor, she works at HMS, she said there were a lot of labs there that she's like, this would be so cool to use on our stuff. I don't know exactly what they're doing, but I don't know how fast we'd be able to get those images, but
You can do this on the bacteria data. Right, so Ramit, you have all the bacteria data set, right? And you guys have already sort of like put in some of the TSME and UMAP type things. Residual, not residual. Yes, it's time also here.
Yes, Dushan.
Yeah. So you are using UMAP, right?
Yeah.
Visualization.
Yeah, the ones I visualize just use UMAP. Yeah, but TSN is also like it's also possible. I think it's just like I didn't use that yet.
Yeah, but the thing is, there's no object detector, right? That part is done beforehand.
Yeah.
So where are they? Yeah. Yes. Did you say something?
Oh no, I didn't understand what you asked, Dushan.
Oh, so in this case, like we have like bigger images where multiple cells are. So the tool is set up like you have an object detector at the beginning of the pipeline, and then the visualizations are done on that image, right? So for example, the image you are seeing has like some 3,000 cells.
Oh.
The TSN is shown on that image, right?
Right. So in the bacteria data set, it is already like segmented and we only have like single cell images.
Yeah. In that case, it's probably not good data set. Alright, let's think about it a bit. I think like we might have some data that is probably in the same format as this. Yeah, but for now we can just focus on the H2X. Alright, sounds good.
mentioned that you're really creating the model running part with image as well. So I'm curious, like, how would that be like invoked, like, with the Python 5 on the system to get that? Like, just curious about it.
Yeah, so that kind of with the packaging and deploying it, that is something to consider because it either runs on the assumption that they do have Python or it would have to somehow package Python with it. But how it's running right now is it has the Python scripts. Here, I can kind of show you, like we have these Python scripts kind of in the same directory right here. So like run TSNE and run inference. And then let's see, down here when it says we like run TSNE and has the script and then has like the Python. So maybe they'd have to like input their Python package because this is always, I mean, I guess we could package the virtual environment as well. That might be the easiest thing to do. And then it will just like run the script from the Java script. So it just runs it like as like command line command. So. Any other comments?
But I think one of the, I'm thinking like, can we do a little bit more science, like in the project? Yeah, this is very engineering, right? It's good, but like thinking that there is a way to like integrate a little bit of science as well. So one thing we can do is, you were there in Mariel's talk, right?
Yes, yeah.
So she like her talk was on like how to do this fishery
Yeah,
statistics.
yes.
But it is basically like a method to analyze the causality.
Okay.
And the extra example she showed is sort of like in the same regime, right? So if you remember, example is basically like there are 17 individuals that they ran a test on. And then the readout is like this 500,000 base pair, like one readout. So you have 34 of them. And it is, you can draw parallels to this experiment, right? So in this experiment, there are a few types of mice. Let's say for simplicity, there are two types of experiment condition. There's the let's take one type of mouse model, and then some are exposed to the toxin and some are not. And then you basically like run this experiment. You sacrifice the mouse and then check in the tissue and then like do this staining and then basically get rid of, right? So for every animal, you have these large, like thousands of data points from every cell. And then you do a quantification here, right? So you can treat these features that you get out of the TSNE as like your readouts basically. So whatever the size of that feature. And now that is similar to what Mariabel gets out of like an experiment where you get this 500,000 readout. So then if we can somehow use a method to just build a test statistics on it, statistic on it.
Yeah.
So basically the idea is like you have this very large high dimensional representation for every animal. And we need to somehow use some model to basically convert that into a scalar.
Okay.
So that scalar is supposed to be whatever the number that gives you an idea of how much that treatment costs this readout. Right, and then we do that Fisherian statistic that I still don't understand, but we can talk to Mariabel and we can do that and then come to a conclusion saying whether that actually had an effect or how much is the p-value.
That's a really good idea.
I think that would be a nice way to wrap up everything.
Yeah.
Yeah, sounds good. Yeah, starting good. Do you have something?
Yeah, I just, I'm in the middle of the road right now, but I typed it, what I heard, from what I heard. So it's regarding like the choice of the parameters. for T-SNE, and also like the choice of T-SNE by itself. Like I don't know whether it's being too mathematical here for depending on the application, but if there was an option, or because now we are making things interactive, for the person to change those hyperparameters as well, maybe that would be nice. Or like if there was a separate study without giving the option for the person to change the parameters of the T-SNE, or if we could like say, okay, this is the most intuitive visualization that gives like clear separation of features in the high dimensions, or like some analysis in that side regarding the selection of this particular version of T-SNE.
When Jonathan built the TSNA visualizer that I kind of base this on, he did have those options. So I think that's actually a really good idea to maybe like before the TSNE is run, have some sort of place where you can choose to modify the input parameters. I think that's definitely something we should add.
Yeah, that's nice. Yes.
Yeah, I think I agree. So that way, like you also get some idea what actually what the DSN is doing, and then what happens when you tweak these knobs.
Yeah.
And it will also be interesting for the user.
Yeah, I think that's a really good idea, and that wouldn't be hard to implement.